Photosynthesis is the process by which plants capture light energy and convert it into chemical energy, forming the foundation of nearly all life on Earth. This intricate series of reactions not only drives the global carbon cycle, but also sustains the growth of crops that feed humanity. However, the natural efficiency of photosynthesis is far from optimal, especially under fluctuating light conditions commonly found in agricultural fields. Enhancing this process offers a promising strategy to improve crop productivity. Such advances are crucial, as current yield trends fall short of the gains needed to ensure global food security in the coming decades. In the dynamic light environments of agricultural fields, leaves are frequently exposed to rapid shifts between high and low light caused by moving clouds and the shading effects of neighboring foliage. To protect themselves from the damaging effects of excess light, plants activate photoprotective mechanisms that dissipate surplus absorbed energy as heat. This process, known as non-photochemical quenching of chlorophyll fluorescence, is vital for preventing photo damage. However, it comes at a cost. When a sunlit leaf transitions into shade, the photoprotective mechanisms do not instantly switch off. Instead, the dissipation of absorbed light energy as heat continues for several minutes, during which time photosynthetic efficiency remains suboptimal. This lag in recovery from photoprotection results in a significant reduction in potential carbon assimilation. Model simulations have indicated that these inefficiencies can reduce crop productivity by 7.5% to 30%, depending on canopy structure and environmental conditions. One of the key contributors to this lag is the slow relaxation of the xanthophyll cycle, a process that regulates the conversion of specific pigments involved in energy dissipation. Additionally, the conformational dynamics of light harvesting complexes within photosystem II further modulate the rate at which photoprotective states are reversed. Recognizing this limitation, researchers have proposed accelerating the relaxation of photoprotective mechanisms as a promising strategy to improve photosynthetic efficiency and, ultimately, crop yield. Genetic engineering approaches targeting components of the xanthophyll cycle and structural proteins of the photosynthetic apparatus have shown potential in enhancing the speed of recovery from photoprotection. The 2016 and 2022 studies by Kromdijk et al. And D'Souza et al. Explore this concept in detail. The first, improving photosynthesis and crop productivity by accelerating recovery from photoprotection, investigates the bioengineering of tobacco plants to accelerate photoprotective recovery. The second, soybean photosynthesis and crop yield are improved by accelerating recovery from photoprotection, examines whether similar modifications can translate into tangible yield gains in a major food crop, soybean. Together, these studies provide critical insights into the feasibility of optimizing photosynthesis to meet the global need for higher crop productivity. To test whether accelerating recovery from photoprotection can improve photosynthesis and crop productivity, Kromdijk and colleagues conducted a pioneering study in tobacco plants. They used genetic engineering to simultaneously enhance three components involved in photoprotection dynamics: the enzymes that drive the conversion and reconversion of pigments in the xanthophyll cycle, and a protein that modulates the conformational state of light harvesting complexes. Specifically, they introduced into tobacco plants the genes for the enzymes violaxanthin deepoxidase and zeaxanthin epoxidase, along with the gene for the photosystem II subunit S protein, all derived from Arabidopsis thaliana. This was intended to accelerate both the induction and relaxation phases of photoprotective energy dissipation. The engineered plants were first tested to confirm increased levels of the targeted proteins. Compared to unmodified plants, transgenic lines showed a 10-fold increase in the enzyme violaxanthin deepoxidase, a 6-fold increase in the enzyme zeaxanthin epoxidase, and a 3 to 4-fold increase in the photosystem II subunit S protein. The researchers then examined whether these changes affected the speed of recovery from photoprotection. They exposed the plants to alternating high and low light conditions and measured how quickly photoprotective dissipation declined when the light intensity dropped. The engineered plants displayed much faster relaxation of photoprotection, with the slower component of recovery reduced from approximately 2,680 seconds in unmodified plants to around 750 seconds in transgenic lines. Next, they assess the impact on carbon dioxide assimilation. After a transition from high to low light, engineered plants recovered their rate of carbon dioxide fixation more rapidly than controls, showing an average increase of 9% in this phase of adjustment. The study also evaluated overall efficiency under simulated fluctuating light. In engineered plants, the quantum efficiency of carbon dioxide fixation under fluctuating light was approximately 11% higher than in unmodified plants. Notably, this improvement translated to the field. Tobacco plants with accelerated recovery from photoprotection achieved an increase of 14 to 20% in total dry biomass under real agricultural conditions. Overall, this work provided compelling proof that enhancing the dynamics of photoprotection can substantially improve photosynthetic efficiency and biomass accumulation in crop plants. Building on the promising results obtained in tobacco, De Souza and colleagues sought to determine whether accelerating recovery from photoprotection could also improve yield in a major food crop. For this purpose, they selected soybean, one of the world's most important grain crops and the single largest source of vegetable protein globally. The researchers genetically engineered soybean plants to express the same three components used in the earlier tobacco study. The enzymes violexanthin D epicidase and zeaxanthin epicidase and the photosystem II subunit, S protein, all derived from Arabidopsis thaliana. The resulting modified plants were tested over 2 growing seasons in field conditions in Illinois, USA. The team confirmed that the transgenic soybean plants produced the target proteins at various stages of development, although expression levels varied among different genetic lines. To test the effect of the modification, they first measured how quickly photoprotective energy dissipation relaxed after a transition from high to low light. In both years, the engineered plants displayed significantly faster relaxation of photoprotection compared to unmodified soybeans. Specifically, in 2020, the engineered lines showed a reduction of 11 to 23% in residual photoprotective dissipation between 90 and 300 seconds after the transition to shade. In 2021, the characteristic time constants for the fast and medium phases of recovery were 37 to 67% shorter in transgenic lines. Importantly, this acceleration improved photosynthetic efficiency under fluctuating light. In 2020, the quantum efficiency of carbon dioxide assimilation in engineered soybeans was 1 to 10% higher than in unmodified plants under fluctuating light conditions. Similarly, the efficiency of electron transport through photosystem II increased by 2.5 to 11.5% in engineered plants. The crucial test, however, was whether these improvements would translate into higher seed yield. In the 2020 field trials, 5 out of 8 engineered soybean lines produced a significantly higher seed yield, with increases averaging 24.5% compared to unmodified plants, and reaching as high as 33% in the best-performing line. Notably, these yield gains were achieved without altering the protein or oil content of the seeds. Thus, the study provided strong evidence that enhancing recovery from photoprotection can improve both photosynthetic efficiency and agricultural productivity in a key food crop. Together, the studies by Kromdijk et al. And De Souza et al. Provide compelling evidence that accelerating recovery from photoprotection represents a powerful and broadly applicable strategy to enhance photosynthetic efficiency and increase crop productivity. Kromdijk and colleagues demonstrated that by simultaneously enhancing the dynamics of pigment conversion in the xanthophyll cycle and the flexibility of the photosynthetic antenna, plants could more rapidly adjust to fluctuating light conditions. Their findings in tobacco showed that this faster relaxation of photoprotection enabled more efficient carbon assimilation during transitions from high to low light, a frequent occurrence in dense crop canopies. Importantly, this translated into significantly increased biomass production under real field conditions, establishing a proof of concept for the approach. The study also provided valuable mechanistic insights. The accelerated relaxation was achieved through a coordinated modification of both pigment cycle enzymes and structural proteins, revealing that a balanced manipulation of multiple components is critical to optimizing the dynamic response of photoprotection. Moreover, the engineered plants maintained robust protection against photodamage, demonstrating that it is possible to improve photosynthetic performance without compromising resilience to excess light. D'Souza and colleagues extended these findings into an agronomically relevant context by applying the same genetic strategy to soybean, a major food and feed crop. Their work confirmed that the approach is transferable across species and functional even in more complex crop systems. Engineered soybeans displayed faster photoprotective recovery and increased photosynthetic efficiency under fluctuating light, consistent with the earlier tobacco results. Crucially, the improved efficiency translated into higher seed yield under field conditions, addressing a key criticism that photosynthetic enhancements might not translate into greater harvestable output. The engineered soybeans also maintain seed quality, with no detrimental changes in protein or oil content. The study further highlighted that field conditions, including light dynamics and canopy structure, strongly influence the potential yield benefits, an important consideration for future application. Taken together, these studies underscored the importance of targeting dynamic limitations in photosynthesis, not just its maximum capacity, to improve crop performance. They also illustrate how a deep understanding of plant physiological mechanisms can be translated into practical innovations for agriculture. As global food demand grows and climate variability increases, such strategies offer promising pathways to achieving sustainable gains in crop productivity, helping to secure food supplies for the future.